Hi everyone, this class is about flame photometry and flourimetry. So these are the two types of photometry. First, we'll see the introduction part of this flame photometry. This is an analytical instrument used for the quantitative analysis of sodium, potassium, calcium and lithium in biological fluids. It's based on measurement of intensity of light emitted when a metallic element introduced into a flame, and the sodium, potassium, calcium and lithium have the properties of emitting a light of the characteristic wavelength. So based on their wavelength, these ions which emits a light of the particular element when sprayed into a flame. So the equipment consists of an atomizer which draws a sample solution and a compressor which pumps air at high pressure. It is fed into the flame and the flame will be blue if the sample contains only distilled water. When the serum sample is introduced, the flame acquires the particular color. So this is the flame photometry instrumentalization, as it consists of atomizer, slits, monochromator light and the detector, and to measure it's a meter. So here also we can see the instrumentalization as a left side, flame, lens, filter, photodetector, display, and the right for the instrument to measure. So basically the atoms of many metallic elements, when given sufficient energy such as that supplied by a hot flame, will emit this energy as a wavelength characteristic for the element alkali metals, which is easy to excite. So the light intensity of emitted light will be proportional to the number of atoms. So the principle of flame photometry includes when a solution of a metallic salt is sprayed on a flame, then the fine droplets form and evaporates, leaving behind fine residues which are converted to neutral atoms. So the atoms of elements subjected to a hot flame will be specific quantum of thermal energy absorbed by the orbital of electrons of an atom and become unstable at a high energy level and release energy as a photons of particular wavelength, and it gets changed back to the ground state. So the energy released in form of a light spectra is characteristic for the each element. So the timings of a electron gets down to the ground state. From the excited state, it emits a light. The emission wavelength of various metal ions will be as the table follows. Element as sodium, potassium, barium, calcium and lithium, and their emission of a wavelength is listed, and what are the flame color as yellow, violet, lime green, orange and red. So once again, the components of a flame photometer includes flame atomizer, monochromator, detector and the readout meter. So the flame atomizer, the component of a flame photometry will be atomization of a sample and source of thermal energy, and it's composed of nebulizer and burner. So nebulizer, as an oxidant flows, it withdraws the sample from the capillary in very fine droplets and mixed in the pre-mixing chamber with the fuel gas and then passes to the burner for the heat and atomization. So most commonly used atomizers are total consumption burner or laminar flow burner. So as a burner or flame, the heat of the flame evaporates the water and vaporizes the substances, which is converted into atomic state. So here the flame provides heat energy. The heat of the flame evaporates the water and vaporizes the substance. And next, the monochromator, or also called as filter. Monochromatic light is obtained by means of filter. The light of particular wavelength emitted by the electron passes through the suitable filter. So, single color of a light or monochromatic light is obtained by this filter. As for sodium, it has 580 nanometer and the potassium 766 nanometer. Next is a phototube or detector. As a detector is a phototube which converts the light energy into electrical energy. So the galvanometer, this electrical energy produced by the detector is measured by galvanometer and the value will be read out in the display. And some limitations of a flame photometry are, as the number of excited atoms in flame is very small, alkali metals can be particularly determined, and it needs perfect control of flame temperature also. And interference by other element is not easy to be eliminated. The heavy transition metals, the number of absorption and emission lines also enormous, and the spectra are complex. So the applications of flame photometer includes, the technique is wisely used in the clinical laboratory to determine the concentration of a sodium and potassium in biological fluids like serum, urine and sweat, and the lithium levels also can be measured in connection with the therapeutic use of lithium salts in the treatment of psychiatric disorders. And also it's more simple and less time consuming compared to the colorimetry. So it includes flame photometry description. And the next we'll see flourimetry as a subtopics, principal, instrument and applications. So the flourimetry, it's a technique in which the absorbed light is re-emitted. The emitted light can be used to quantitatively determine the light emitting compound. So here the fluorescent light is the result of the absorbance of a photon of radiant energy by a molecule. So the molecule upon absorbing energy, which leads to the excited and has a higher energy level, returns the excessive energy which is lost as an ejected photon. So the result is fluorescence. So the fluorescence, it's not exhibited by all compounds absorbing energy, is different for different compounds. So quantization of a substance by using the property of a fluorescence is known as fluorometry. So the fluorometer which measures the fluorescence of a compound, the components are light source, monochromator and detector. So here the instrumentation includes source of light, the filter, monochromators, sample cells and the detectors. First we'll see the source of light as mercury vapor lamp or xenon arc lamp or tungsten lamp. These are the source of light used for the flourimetry, where the mercury vapor at high pressure gives intense lines on continuous background about 350 nanometer, and the low pressure mercury vapors gives an additional line at 254 nanometer, which is used in filter flourimeter. So xenon arc lamp which gives a more intense radiation than the mercury lamp, so it's used in the spectrophlorimeter. And tungsten lamp, if excitation has to be done in visible region, this can be used. Next is the filters and monochromators, that is filters, these are the nothing but optical filters, works on the principle of absorption of unwanted light and transmitting the required wavelength of a light. In inexpensive instruments, flourimeter, primary filter, the secondary filter are present. And the sample cells, these are meant for holding liquid samples. So if we need to, the sample has to be checked with the flourimeter, the sample cells has to be there as they are made up of quartz and can have various shapes. example cylindrical or rectangular or etc. And the detectors will be photometric detectors are used. They are barrier layer cell or photovoltaic cells and photomultiplier cells. So this is the picturization of an instrument, as when the energy of a particular wavelength is passed by the scintillator, the ionization track is present, and the low energy photons, high energy photons all get together by the and leads to the photocathode and releases primary electron and leads to the secondary electron, which focused on the focusing electrode which is present in the instrument, and as it forms a multiplayer tubes. Multiple electrons are formed by the PMT, that is photomultiplier tube, which consists anode, dynode, and which is connected to the pins. So the factors affecting the fluorescent signal will be solvent which we are using, and the pH, the temperature of an instrument, absorbance of a solution, and presence of interfering substances in it. Here the standardization is not usually done by an absolute procedure, because fluorescence varies depending upon the intensity of a light of incident on the sample and the amount of light pursued by the detector, which is controlled by the slits, and the band width of the light analyzed, and the efficiency of the detector. So these are the four dependent factors for the fluorometer, and relative fluorescence is used for the measurement. And here the fluorescence attenuation assays use the inverse quantitation relationship between fluorescence intensity and the amount of light absorbed by the solution. So time delayed fluorescence would be the method used to improve sensitivity of fluorescence technique. The usual fluorescence emission time is extended by using these compounds with a long fluorescence lifetime. So the advantage as improved specificity and sensitivity, and the disadvantage will be requires specialized instrument with separate steps, as chelates cannot be directly measured by the in body fluids. So the applications of a flourimetry includes, it's a methods not useful in qualitative analysis but will be using in quantitative analysis, and the fluorescence is the most sensitive analytical technique which has a more sensitivity and specificity. Detection studies will increase the development of fluorescence field. And flourimetry is widely used by the dairy industries to verify whether the pasteurization has been successful. So this ends with the completion of a photometry types, that is flame photometry and photometry. Thank you.